Hello everyone, welcome back to my YouTube channel. So as we are dealing with the immunology series, so first we have discussed about the what is immunology, then what is innate immune system, what is immune response and what is innate immune system, and today we'll be discussing these inflammatory barriers and we'll be discussing the adaptive immunity. So under adaptive immunity there are two types of immunity, humoral immunity and cell mediated immunity. So for that, I'll be separating the video in different parts. So today we'll be continuing with this inflammatory barriers and humoral immunity. Okay, so let's start. So first, it is the inflammatory barriers. So if a pathogen wants to enter our body through a tissue injury or a injury by anything, by it can happen by anything like see in the picture you can see one damage occurs in the tissue. So that particle or that thing is there that is inserted in our tissue. So this damage can leads to the entry, or it promotes the entry of pathogens in the tissue. So in the second picture also you can see that the after removing that thing from the tissue, the small microorganisms are present over there. So this leads to the inflammation. So now how this leads to the inflammation and what is the mechanism is going over there, we'll be discussing in different steps. So first is the vasodilation step. So when the tissue damage occurs, then the engorgement of the capillary network occurs. That is responsible for the tissue redness and rise in tissue temperature, also called erythema. So after entering of that the particle or the thing inside our tissue, or penetrating in the thing inside our tissue, first step is vasodilation. It enters and the engorgement of the capillary network, it occurs, and then it leads to the tissue redness and a rise in the tissue temperature. When this kind of things happen, if you touch that place, you can feel the hotness or the temperature, rise in the temperature on the of that area. That situation is called as the erythema situation. So next, increase in the capillary permeability or occurs, that mediates the release of the exudates and leads to the swelling of that that is called as the edema. That swelling is called as the edema. So we all know that if that kind of injury occurs, swelling occurs. Depends on the cut or the affected area part. The inflammation or this swelling depends, like how, whether it is a primary injury or a secondary injury. Based on that, the swelling, the proportionality of the how the swelling occurs, that is different based on the different condition, but that swelling is called as the edema, that you have to remember. And this is the second step, that is the increasing in the capillary permeability, and that mediates the release of the exudates and leads to the swelling of the edema. So that is the second step. So now then phagocytes come to the capillary wall, which is called the margination, and then this cells extravasate through the cell layer, which is called the extravasation vasation. After that, they will go and bind to the pathogens that are being released in the tissue, damage and neutralize them. So what is going on? These phagocytes, now it will. So we all know that phagocytes is a primary component of the innate immune response. So due to that, these phagocytes will, when this damage in the tissue occurs, these phagocytes is the first cells, like not the first cell is neutrophils, but like these phagocytes will try to engulf that pathogen and delete that pathogen from our body, no. So for that reason, these phagocytes will come and it will invaginate through the cell linings, and it will cross the barrier, that cell lining barrier, and it will go and it will engulf that pathogen on the inflammatory site or the inflammation site, so that so that it can easily engulf that pathogen, it can degrade that pathogen by the phagocytosis process. Or if that macrophage cannot be able to degrade that pathogen, it will go and it will present that pathogen to the T cells and the B cells, and that we are going to discuss in the next slides. That is about the that adaptive immunities and all. So but this inflammatory barriers, so three step. First is the vasodilation step, next is the edema, that is the what is that, swelling, and last step it is the deletion of the pathogen, the encounter of the phagocytes to of the like it, that phagocytes will encounter these pathogens and it will try to degrade that pathogen. So that is about the inflammatory barriers. So next, it is the adaptive immunity. So this adaptive immunity is a very important topic. So guys, listen properly, and if you find this is an informative, then please note down the points. So first, what is adaptive immunity? So adaptive immunity is a specific type of immunity that is capable of eliminating the foreign pathogens with more specificity. So I'm telling again that adaptive immunity is a specific type of immunity that is capable of eliminating the foreign pathogens with more specificity. So that innate immune response is a less specificity. It is a primary defense mechanism of this of the body. That is the innate immune response. But the adaptive immunity is a more specific. So these cells will properly go encounter to that pathogen and it will degrade it off, it will delete it off completely. So now attributes of the adaptive immune response are antigenic specificity, diversity, immunological memory, self and non-self recognition. So one of the important part is the immunological memory. So what is that immunological memory? So first, this cell, this under the adaptive immune response, the B cells and the T cells, these comes under the adaptive immunity. So these B cells and the T cells, no, they can form the memory cells. So what is happening? First, this is the B cell. So this B cell will encounter a pathogen. So this is the first time it is encountering a pathogen. So after this encountering, some first time it will need some time to degrade that pathogen or to build an immunity against that pathogen. But what is happening in the next stage, again the same pathogen when attacks the body, the after this first encountering of that B cell or the T cell to that pathogen, no, this B cell and the T cell release some memory cells, so that that cells will have the antibody specific to that antigen only. So next time when that antigen comes in the body for to attack the body, then that memory cells will go and it will encounter that antigen quickly and it will degrade very fast. That is the role of the memory cells. And based on this application only, this memory cell concept only, the vaccines are produced. Like different vaccines are, like recently this COVID vaccines you say, or any vaccines like subunit vaccines. So what they are doing, they're taking a part of a pathogen, and in the subunit vaccine, they're taking a part of the pathogen and they're injecting that pathogen part in the body as a form of vaccination. So after when we took that vaccination, our body, in the first time our body might feel some say for suppose that COVID vaccine, no, so after taking first dose, everybody or most of them are feeling some cold or some similar type of COVID symptoms, but it is not that much, but little bit similar to the COVID, like common cold or flu kind of thing. But why this is happening? Because the scientists, they're injecting a particular subunit of the virus in our body so that our body can generate antibody against that. So when that actual virus comes in our body, we have already the antibody against that antigen. So that antibody, what this will do, this will go and this will bind to that antigen, and it will quickly degrade that antigen or degrade that threat or remove the threat from our body. So that is the with the help of the immunological memory only. So that is the concept of any like the subunit vaccines or any vaccines, most of the vaccines actually. So now, so there are two types of the immune responses involved in it, as I already said, that the humoral immune response and the cell mediated immune response. So first, today we'll discuss the humoral immune response. So in this humoral immune response, it is nothing but the B cell response. So now this B cell, you say that what is that B cells and all. Since these B cells are the nothing, it is the immune cells, and these are generally generated from the bone marrow, and this is they are matured in the bone marrow only. That's why it is called as the B cells, and there's some theories are there. Okay, but the main mechanism today we'll discuss, because that is most important thing. So first, involvement of the B cells plays an important role in this immune responses. B cells can act as both pathogen, antigen presenting cells, as well as active lymphocytes. So this antigen presenting cell, guys, you remember from the far, if you don't know what is antigen presenting cell and what is the innate immune response, you can go back to my previous video, you can watch that video. Again, I'll be repeating this thing. This antigen presenting cell, it is nothing but that the and this particular type of cell, those will present the antigen or the pathogen to the T cell, so for the better immune response. So this B cell can also act as an antigen presenting cell. Apart from that, it is also having a role that it can directly go and encounter pathogen and degrade the pathogen. Apart from that, it can also act as an antigen presenting cell. See, B cells can kill pathogen in both T dependent as well as T independent manner. So after activation of the B cell, if it follows T independent mechanism, then it will directly bind onto the surface of the pathogen and can destroy the pathogen. If they are following T dependent mechanism, then the B cell acts as antigen presenting cell. See, this T, if they are following the T dependent mechanism, like T means T cell dependent. Okay, so what they are doing, they're taking that antigen or the pathogen to their surface, and through the MHC2 complex, that is a major histocompatibility complex, they replace they represent the pathogen or the antigen to the T cell. So that is how they are doing the process. They are acting as an antigen presenting cell over there. So now there are different co-stimulator, there are different cytokines involved in this process for this activation of the B cells, and the B cell and the T cell interactions we'll be seeing in the next slide also. See, after activation of the B cell, it will proliferate and produce two types of the cells, that is one plasma cells, from where the mature antibodies will release later, and the memory B cell, the memory cells. See, the memory B cell will be activated in case of secondary infection by the same pathogen. As I have already said, this memory B cells or the memory T cells, these are the very important topics that you have to remember. So in the next slide, see, this is a very important slide. See, this is the B cell receptor. So this B cell receptor, also known as an membrane bound immunoglobulin. Okay, this is a typical immunoglobulin or the antibody structure. You can find this heavy chain, light chain, variable, this constant region, and then this antigen binding region, hinge region, everything you'll find this FC fragment, the fragment of FAB, that is the fragment of the antigen binding region, everything you'll find over here. But what are the additional things you are looking here, or you get at that? See, this IGF, this IG alpha and IG beta, this is an additional thing. So B cell receptor has a similar structure as an antibody or immunoglobulins, except that B cell receptor has an extra signal domain at the C terminal region that helps in the attachment of the B cell receptor to the membrane, and it consists of a transmembrane and a cytoplasmic domain segment. But the points to remember here is you can see this cytoplasmic domain is a very small domain. So if even if the antigen will bind to that B cell receptor, that signal, no, that signal needs to go to the nucleus, and that is most important thing, that the signal needs to go to the nucleus. Then only cell will function properly. Without signal, it will not function. So for that, we have what? We have Ig alpha and Ig beta. So this is an heterodimer receptor. Okay, this is an heterodimer receptor, and this is having a single Ig subunit on the top. So what they'll do, they're like, they have a large cytoplasmic domain, as you can see in the photo also. So due to this large cytoplasmic domain, so this will transfer the signal to the, this helps in the transferring of the signal after antigen binding to the nucleus, so that the cell will function properly. So this is this Ig alpha and Ig beta, these are the glycoprotein transmembrane molecule. Extracellular portion consists of a single Ig like domain. It is only expressed in the B cell. Clustering of this B cell receptor activates the multiple Ig alpha, Ig beta molecules and initiate the signaling process. So antigen will bind, it will generate the signal, and this then that signal will be transferred by this Ig alpha, Ig beta to the cell nucleus, and after that cell will decide whether that B cell will go for the T independent pathway or the T cell independent pathway or the T cell dependent pathway. So there are two pathways, that is a T cell dependent, T cell independent pathway, and there are different signalings in this pathways that we are going to see in the next slides. So in the next slide, as you can see, this is a T cell independent B cell activation pathway. So this T cell independent B cell activation pathway, when this will occur, see, this will happen when an antigen will be having the repetitive identical epitopes. So when this antigen is having a similar type of the epitopes is the region where the, it is the antigenic region where an antibody can go and it will bind. So this that antigenic region is called as the epitope region. So if this antigen is having multiple or multiple repetitive identical epitope region, so this T independent pathway will occur over there. So what is the pathway going on here? See, the B cell recognizes and bind to the antigen, and this is the first signal for the B cell activation. So T independent B cell activation, what is going on here? First B cell will encounter in the with the multiple say identical epitopes region in the antigen. So after that, toll like receptor, so TLR is a short form. So toll-like receptor that can recognize various microbial surface protein, it will also bind to that. This receptor molecule, this will also binds to the cells, and then this recognition and binding gives the second signal. So binding of the toll-like receptor, this provide the second signal. So after activation, plasma cells will be produced, but memory cell will not produce. That is the second important thing. See, this after activation, so everything, first signal comes, second signal comes, B cell gets activated, and the plasma cells is produced. From that plasma cells, I'll tell that the plasma cell is the cell, from that the antibody will be released, and this is the first encounter, no? So first what will happen, this after the B cell activation, plasma cells will be released, and those plasma cells will produce the antibodies, and that antibodies only, this will go and this will bind to the antigen and it will degrade that antigen. In the picture also you can see this, after encountering this B cell with the antigenic cell, this T cell T cell independent B cell activation, this B cell will be activated, it will go, it will mature, and it will mature into a plasma cell, and from that plasma cell the antibodies will be released. So that is the concept, but the memory cells here, it will not produce in this pathway, in this T cell independent B cell activation pathway. Why? For the activation of these or for the generation of the memory cell, T cell interaction should be present or should be there. Without having T cell interaction with the B cell, like B cell T cell interaction, this B cell cannot be able to produce the memory cells or the memory B cells. It will delete off the antigen, but if the antigen is attacking the body second time, this body again it will follow the first pathway only and not the more effective pathway, that is the T dependent B cell activation pathway. So T dependent B cell activation pathway, it is the most effective pathway, or at least the effective pathway than the T cell independent B cell activation pathway. This antibody, this IgM antibody, those are generally produced by the B cell activation or the T cell independent B cell activation pathway. So that is another important thing to remember. So moving on to our next slide. So now we'll discuss about the T dependent B cell activation pathway. So in this pathway, what is going on? It's a bit complex picture, you can say, but trust me, if you understand the concept, it will be very easy. So in this pathway, first, there are three signal pathway. Again, first signal, it is the B cell with the antigen interaction. Second signal is the co-stimulators. There are different co-stimulators are present, that is the CD40, CD40 ligand, CD40 receptor interactions, and then the third is the different interleukin interaction that will lead to the activation of the B cell. So, and in this pathway, again, I'm saying that the B cell and the T cell interaction is there. Because of that only, the memory B cells will be produced. So, first, antigen and B cell receptor interaction is there. So, this B cell acts as an antigen presenting cell over here or in this pathway. So, this B cell will process the antigen first. After processing this antigen, this will present this antigen to the T cell. So after processing of that antigen only, so this antigen then this through this B cell will present this antigen through the this like MHC molecule, that is the major histocompatibility complex class 2 molecules to the T cell receptor. So this interaction will mediate the activation of the B cell and the T cell, and this second interaction is the CD40 ligand and the CD40 receptor interaction. CD40 ligand is present in the T cell and CD40 receptor it is present in the B cell. So this ligand and the receptor interaction mediates the more efficiency in binding of the B cell and the T cell and more activation of this pathway. There are several other co-stimulator molecules are also present, and like that will lead to the activation of these two cells. And after that, this different interleukin, you can see is this interleukin 21. It is it is helping in the activation of the T cell and B cell also. This in the B cell also gets activated. Different interleukin 6 molecule is also binding to the T cell, because T cell will have interleukin 6 receptor. So these are the three segments of this T dependent B cell activation. Through this T cell, B cell gets activated. So this is the pathway. So if the humoral immunity concept, it's asked, you have to explain this B cell, what is the humoral immunity, and then what is the B cell receptor, and after that these two activation pathways, the T dependent B cell activation and the T independent B cell activation. So these and all things you have to explain in the under this topic. So that's it, everyone. Thank you for listening to my video, and if you like this video, then do subscribe, do like, and do share this channel, and thank you.